Thank you for the introduction and thank you for having me today. It's my pleasure to be here to discuss the benefits of Hyperion IMC in the study of the tumor ecosystem. Today I will divide my talk into three main chapters. First, I will briefly discuss about the need for these high parameter imaging studies to characterize the tumor heterogeneity in a disease-relevant manner. Then I will briefly show you the workflow we use in our lab to perform these high parameter imaging studies. And then the last part of the talk will be to cover a typically controversial issue within digital pathology as cell segmentation is. Nonetheless, this is a crucial step in order to analyze your data and get what is normally referred as single cell studies in tissue. I believe I have half an hour for this talk, so without further ado, let's get started. So first, what we should address is what many people ask. What is the real value of high parameter imaging studies and why they are important, not only in basic research, but also in clinical settings? So I would like to explain this topic using this nice scheme. So I want you to focus in the second and third column here, biomarker analysis and stratification. So what biomarker analysis allows you is to stratify your patients. And why you need to stratify your patients? You need to stratify your patients because you need to assign treatments based on the biomarkers or the set of biomarkers each of these patients present. This is part of what we call precision medicine. You decide treatments based on the likelihood for these treatments to be effective in patients. And the way to do this approach, or the way to decide what might be the best treatment for a patient, is by analyzing the biomarkers and set of biomarkers each patient presents. So this is for cancer. And as you can see here, depending on the type of cancer you may have, you may look at different biomarkers. Of course, this is not an extensive list of biomarkers, but what I want you to look at is this column here. So most of these biomarkers, if not all of these biomarkers, have to be assessed in tumors. So it's not enough to assess the peripheral blood of the patient. So you need to look at the markers inside, so in the tissue. And why you need to do so. So let's try to understand or make sense of this this point. So this is what happens when you look at many parameters using, for example, peripheral blood. So you have your data, you sort your data, you arrange your data, and then you present it visually. And why you do that? Well, the first thing is you do a qualitative analysis. So you said, you can say, if you have or not some markers for X population. And then once you present this marker in a kind of understandable, visually understandable manner, you can even quantify this marker. You can say you have more red than yellow and more yellow than white, and then you have more white than blue. But what Hyperion IMC allows you is to go one step further, and is to explain all this data with the story. So now you know that you don't only have, let's say, determined amount of blue. You know that the blue here are the roof of the house. So you put all this data in context. So normally multiplex suspension allows you to go until the data range. Then there are very good and very well-known algorithm like UMAP or like TSNE, which allows you to present the data in a kind of, as I mentioned, visually understandable manner. And this is what Hyperion IMC allows you to do, to go, as I mentioned, one step forward and put all of this in context. So let me show you how all of this looks like in real life. So I will show you part of our result. This is still work in progress. But I think it might help you to understand how you put all this data in context. So we are working with two different types of adenocarcinoma. One is breast cancer, and there is CRC, colorectal carcinoma. We are using FFP sections and tissue microarray. I will go back to tissue microarray in a few slides. For this project, we're also using this panel I'm showing here with 21 markers plus a DNA marker. As you can see here, the DNA marker is equivalent to the DAPI in immunohistochemistry. So later in the talk, I will also go back to the approach we use for panel design. This course here are all from the same patient. So this is only one patient sample. And the results are for some of the positive markers we found in this patient. So each of the markers you are seeing here, excuse me, is a very small snapshot, but you have from architectural markers like SMA to immune markers like CD8 or FOXP in this core. So each of the markers has been assigned with a red or green color or pseudo color, and DNA is shown here as blue pseudo color. So what I'm showing here is just how different the results or the interpatient results might be. So you have PDL1 here and you have PK here. The snapshot you are seeing here is a merge of PK and PDL1. As you can see here, these first two patients have a good amount of PDL1 and pankeratin, but other patients you have little to, I would say, none PDL1 and pankeratin expression. You have a lot of stroma here, which is surrounding the tumor, but the tumor has not PK nor PDL1 expressed on it. So, however, as we see here in these results, PDL1 expression does not seem to be related to neither the tumor grade nor the tumor stage, and certainly it doesn't, as it has been shown many years ago. So, certainly the prognostic role of PDL1 in adenocarcinoma, in cancer adenocarcinoma at least, remains unclear. So there is also a debate about the role of macrophages and T cells in the tumor ecosystem and how they interact with cancer cells. Thus, in our work, we have included T cells and macrophages, just as two other examples of the markers you can consider when trying to assess the prognosis and clinical pathological value of some biomarkers. So as you can see here, regardless of the level of PDL1, you have or seem to have many T cells and macrophages invading the tumor or being close to it. So you have a lot of PDL1 here, and you have a lot of macrophages expressed by C6CA in white here, and T cell expressed by the red CD3 here. But you have, for example, another patient with almost no expression of PDL1, but still with a lot of T cells and macrophages. So it seems like PDL1 is not directly correlated with at least a number of macrophages or T cells you have. But there might be another questions you want to answer. For example, is the location important here? I mean, is the location of T cells or the location of macrophages important here? Is or what cells are close to the tumor? And is this special information related to the clinical outcome? So please, don't forget that all this data is meaningless if you don't correlate it with the clinical outcome. So to stratify patient and to decide the best treatment for each of the patients, we postulate that you have to look at all this in context, but you have to look at all of this paired with the clinical outcome for each of the patients. So this is the special results we did for the course I showed you in the previous slide. So what we did was to measure the distance you have between T cells, macrophages, and tumor or tumor cells. So you can see here two different types of plots. we did the proximity analysis within 100 microns and within 200 microns, meaning we asked the software how many cells or what's the percentage of T cells or macrophages within 100 microns to the tumor or from to the tumor. And we did the same using 200 microns as a reference. So the first you can see from these plots are, I would say, two conclusions. The first one is you have much more bars for T cells than for macrophages. And this is because macrophages seem to be more packed than T cells, which are more, let's say, sparse within the cores. And the other conclusion you can get from these two plots is that both macrophages and T cells are no farther than 100, I would say 140, 150 microns, because you don't have cells farther than 100 microns. So all of them seem to be closer to the tumor, at least within 150 microns. But let's see what happens when you zoom into this data. And this is very important. When you zoom into your data, and I think this is very interesting, you have these results in here. And let me walk through this result very briefly. So we found that 99.1% of CD68 are within 100 microns. 97.5% of T cells are within 100 microns. So this is, I would say, quite similar between macrophages and T cells. And then you have that 1.12% of the macrophages are within 10 microns. We are zooming even closer. And then you have the, for T cells, you have that 1.75% of the T cells are within these 10 microns. But this is very interesting. I want you to focus on these results. So 100%, meaning all of the 1.12% of CD68 cells are within 7 to 8 microns from or to the tumor. So we are not talking about 10 microns. We have narrowed the result for T cell within 7 to 8 microns. All the cells are packed here. But you have 0% of CD3 positive cells within 7 to 8 microns, meaning the closest cells, T cells, to the tumor are 9 microns far. And this is very important because there is a lot of debate going around this topic. What is the function of T cells and what is the function of macrophages within the tumor microenvironment? But now we are going a step forward. We are just talking about distances, and these distances may have a clinical implication. So as I mentioned, all these results paired with the clinical outcomes may give you the key to decide the best treatment for your patient. So now in the last part of the talk, I will show you what it takes to go from sample preparation, sample prep, to the results I showed you in the previous slide. So the first you need is obviously the Hyperion imaging system. Hyperion IMC Imaging Mass Cytometry uses the well-known CyTOF technology, in which, as you know, limitations of the fluorescence label antibodies are overcome by using pure metals, as you can see here, that are separated by mass instead of by wavelength. So with an appropriate panel design and appropriate sample optimization, you have to deal with, I would say, little to none spillover and compensation, as many of you may know. So the first step of any experimental design, including Hyperion IMC, is panel design, meaning to design the panel that will be in the antibodies that will be in your panel. So I think it's redundant to mention, but the markers in your panel will depend exclusively on your scientific questions. However, to make things easier, I recommend to start always from a proven foundation instead of starting from scratch. And the foundation I'm talking about is this backbone. This is only a figure, but our panel, our backbone panel is this one. So regardless of the project we are working with, we always work with the same backbone, which is this one in here. So this is actually a panel in which we have 3 different group or set of markers. We have architectural markers like SMA or like Vimentin. We have immune cell markers like CD45, CD4, CD3, CD8, CD68, etc. And then we have tumor markers. We have icadogenin, we have PD-L1, we have Ki67. So this is our backbone. I would say that regardless of the tumor we're working with, we use the same backbone. And then what we do is just to add things around, like you can see here. So these drop-in antibodies are antibodies we have added in open channels we had. So we are looking at not only FOXP3, but we are also looking at LAG3, which, as you may know, is kind of a marker that many people are studying right now, because it might have a kind of predictive value when deciding, as I mentioned in previous slide, the best treatment for each patient. And something you can do is, as you can see here, we had this CD8 labeling another channel, which was 162. So what you can do is just to move things around, because we like another marker in 162. So what we did was to get the purified CD8, the same clone, and then we use the labeling kits Fluidigm provide. The labeling kits have a kind of a very robust protocol. So in 4 or 5 hours, you can label anything. If this anything means IgG, purified IgG without any carrier. So you can label any antibody you want. And this is what we did here and what we did even with PD-L1, because the PD-L1 in the original panel doesn't work for us. So panel design is very flexible. You can just put the clones that work for you and just put them in the available channels you have in your panel. So this is the way we normally in our lab approach to panel design. So remember, use a backbone and then you just build around this backbone. So next step is staining. Basically, staining does not differ from the typical protocols for immunohistochemistry. So they are exactly the same. The tip and tricks I can give you here is, when possible, use what you have here, which is called TMA, tumor or tissue microarray. So as you know, tissue microarray has cylindrical cores, all of them normally from different paraffin donor blocks. So each of these core is a patient, as you can see here. So the benefit of using the TMA is that you can put many patients in only one slide, and then you will stain this slide glass with only one antibody cocktail. And then as Hyperion is all automatic, you will set up the software and it will ablate all these cores and you will walk away. So you can have 70 patients, 72 patients in the same slide, and you walk away and you will have results in less than 3 or 4 days for all these patients. You can even run overnight and you don't need to be there. Just go there once the results are in your hard disk, and then just take it and do data analysis. So again, when possible, my recommendation is use tissue microarray. Once you design your panel and stain your tissue, you have to ablate the tissue using the Hyperion imaging system. So as you know, Hyperion is comprised of 2 units, as you can see here. One is the Hyperion tissue imager, which is here, and there is the excuse me, mass cytometer, which is here. The magnified image you have here shows the ablation chamber of the Hyperion, where you will place your slide glass in order for this to be ablated. So we use slide glass exactly the same as you use for immunohistochemistry. So no special requirements in there. So I don't want to bother you describing in detail the technology. Probably for the time being, it will be enough to say that Hyperion has a UV laser inside, and that this UV laser ablates the tissue at a precise 1 micron, meaning the resolution of the instrument is 1 micron. So the tissue ablated by the laser is then carried to the mass cytometer, and then the ablated tissue is ionized in a high energy argon plasma, and then the resultant ions are brought to the time of flight chamber, where they are measured by a single detector on as you know, on the basis of their mass. This is something important to remember as we enter in the last part of the data analysis, and things start getting interesting here. So once the ions are measured, the way the software uses to reassemble the image is by remembering what was in each of the 1 micron sample that the Hyperion sent to the detector. So the way to record this information is for each of the channel, record the, as you can see here, you have values for each of the markers, but these values are recorded in a determined XY position. So what the software does is, at the end of the process, reconstruct an image based on the information for each of the markers, in each of the XY ablated. So an image that may look like this is what you will have as a result. So based on the information for the 6 markers, for example, the 6 markers that I showed in the previous slide, the software will reconstruct the image. Now, what you have to remember is that even though you are seeing here a perfect image of a tissue with cells positive for PK, for CD3, for CD68, for E-cadherin, for collagen, and you are seeing the DNA positive in here, this is no more than X and Y data. For a software, or I would say from a software standpoint, this is a group of pixels. These are not objects, let alone cells. So your eyes are seeing cells, software eyes are seeing pixels. So the next step in any data analysis workflow is to convert these pixels into cells. And the process of converting pixels into cells is known as cell segmentation. So the last slide of this talk will be to explain the cell segmentation process. I would say the bottom line is there is no perfect cell segmentation tool. All of them have pros and cons. Probably the decision about what tool to use will depend on your scientific questions. One of the data tools that we use in our lab is called HELO. People in the field of digital pathology probably may be familiar with HELO. HELO is a software licensed by Indica Lab. This is a software that works pretty well for us. Is very intuitive if you train the software. And I would say that the sweet spot of this software is when you have, or when you are dealing with many parameters, like the parameters you have when you do Hyperion analysis or Hyperion experiments. So let's see how segmentation works. So there is a lot of debate around the segmentation topic, and but probably one of the points everyone agrees is that cell shape and nucleus shape matter. So it's not the same to segment immune cells than to segment, for instance, spindle shaped cells like the smooth muscle cells. So round cells are very easy to identify and are very easy to segment, while on the other hand, cells that have different shapes are kind of challenging. So what we did in our study was to use this kind of spindle shaped cells to train our software and to see if the software can do a kind of decent job recognizing cells that are not round cells. And these are our results. So as you can see here in A, we have what it looks like to be a cell here. Then we put exactly the same tissue stained with H&E, and we put under the microscope, and we checked that actually this was not a cell, but they were two cells. And we run the same core, the same region of interest in Hyperion, and then after running Hyperion, we put the image in our software. And as you can see here, Hero software did a kind of decent job identifying that this was not one cell, but two cells overlap one each other. And we did the same with another ROI. So we have what it seems to be here only one cell, spindle-shaped cell. But then when you look at this image in the microscope, under the microscope, you can see that there are three or at least two cells. And the software recognizes three cells in there, overlap one each other. So as you can see here, our software did a kind of decent job recognizing cells that are not round cells. So after training our software, what we did was to put our course and base our study on two different groups of markers. The first group of markers are cancer markers, and the other group of markers are immune cell markers. So as you can see here, we run segmentation in our course, and the software did a great job identifying our PK positive cells. We did the same for PD-L1. And then on the other hand, we did the same analysis for immune cells, as I mentioned, and this is only an example showing NK cells. And as you can see here, this is from where we started, and then we run the segmentation tool. And after segmented our course, you can see here that the software perfectly identified our immune cells, in this case, our NK cells. So remember, the first step before going into data analysis is to segment your cells, meaning to transform your cells from what is here, pixels, to what is here, objects or cells. This is what you have to do. And then you have as many downstream analysis option as scientific questions you have. So you can do as we did here. This is called proximity analysis. So we run a proximity analysis, and you can see here the software now is perfectly identifying our PD-L1 positive cells. Then you can put your T cells in here. You can put your macrophages in here. You can put your tumor cells here. Our tumor cells actually are overlapping with PD-L1, so we can take out the PD-L1 cells and see our tumor cells in here. And now you and the software are speaking the same language. You and the software are speaking the language of objects. And then, as I mentioned, we did, well, a lot of analysis. This is only an example where you can see the proximity analysis we run. This is a proximity analysis for CD68 positive cells. This is a proximity analysis for PD-L1 positive cells. And as you can see here, this is a real image we had. We don't have the marker in here. What you have here is what the software recognizes. And as you can see here, I know it's difficult to see, but you have like small dots connected in here, and this is what the software did to detect the cells within, as I showed you in previous slide, within 100 microns and 200 microns. And then you can, again, this is endless. You can create the models you like. What we did was to create models with heat maps where the software recognized where the PD-L1 positive cells are. As you can see here, these are two different patients. This is patient 1 and this is patient 2. So you can see the heat map is completely different based on the location of tumor markers and the location of, well, you are seeing here the T cells and the CD68 positive cells. And then you can move from here and go back to the single cell analysis and recognize where your cells were in this heat map. So the as I mentioned, the amount of analysis you will perform will depend much on your scientific question. But once you segment your cells, the downstream analysis will be almost endless. So I think this is my last slide, and I think I am very close to my 30 minutes. So I will just skip this slide, and I will thank you again for having me here today, and I will be very happy to answer any question you may have. Thank you for your attention.